Okay, guys, so this is going to serve as our kind of overview of the lymphoid or lymphatic system and immunity or the immune system. It's going to be a bit long because I'm going to lump it all together, so just kind of skip around and choose what you need. It should just be an extra resource for what we've covered in class, a little more reinforcement for those topics. Okay, so let's start off. The biggest thing, most important part of this whole slide is this very top part here. Okay, so what is the lymphatic system? So remember that one of the things we learned early in the year is that blood vessels leak, particularly capillaries leak. They're designed to leak. They're supposed to leak, okay? And one of the biggest jobs of the lymphatic system is to return that fluid into the bloodstream. So some of it goes right back into the bloodstream. Some of it gets sucked right back into the capillaries after it leaks. But a lot of it gets collected by the lymphatic system and kind of pumped back in, okay? And we'll kind of show you how it does that down below, okay? The other thing that we do is we're going to kind of filter and clean that along the way. And it's going to house, we're going to house all sorts of immune cells within there to be able to work on that. And you'll hear about lymph nodes and so forth. So let's talk about the pieces, okay? So lymph is actually the fluid. So when the fluid, when the blood plasma leaks out and bathes the fluid, and we collect some of that, it's now referred to as lymph. And it's going to be returned through these lymphatic vessels. So they're kind of like capillaries. They're a little bit larger, closer to veins actually in structure. Most of them are. And they're going to dump everything back into the subclavian veins. So remember, the subclavian veins are those two big veins that kind of that go into that superior vena cava, and they are right underneath the clavicle, right? Subclavian, so they go right into the clavicle and go into, come back from the arms, right? So that's where we're going to dump everything in. Okay. Along the way, we're going to have lymph nodes, and lymph nodes are essentially like checkpoints where they're going to filter and kind of check for infection and so forth. And that's where you hear about like swollen glands. Okay. The thymus, we'll show you that in just a moment. It's this gland that helps train the T cells. You'll hear a lot about T cells in this unit, and that's where they're kind of trained. The spleen is this kind of large organ that's kind of wrapped around the side of the stomach, so it's on the left-hand side around your stomach there, and it's kind of like a large lymph node, but it's the blood flows through it. Okay, so it's a lymph node for your blood, where blood's going to flow into it. It's going to be searched for infections and cleaned up. Okay, and then two random vocab words at the bottom here that we want to make sure we talk about. Pathogen is our general term for anything that can cause disease, so a virus, bacteria, fungus, parasite, any of that kind of stuff. And then antigen at the very bottom. We talked about this with the blood unit. These are protein markers on the outside of cells, and they serve as kind of identification tags and so forth. All right? So let's look at a couple of those things in more detail. So here's a slide that just shows that kind of leaking idea. So here's blood flowing from, red won't work real well there. Let's use some orange, okay? So here's blood flowing from your heart down to a certain area. So let's say this is the middle of your big toe, okay? And here, these little arrows are showing that these are supposed to leak. And all these cells right here are bathed in this fluid that's got all the new oxygen in it and glucose and all the things you need. Some of that fluid goes right back into the capillaries. So some of this goes right back in here, which they don't show very well in this picture. But a lot of it goes into these green, and the green is supposed to indicate the lymphatic vessels. So we're intentionally leaking, bathing the cells, and then some goes back into the blood vessels, but a lot of it goes back into the lymph and starts making its way back up to those subclavian veins to be dumped back in. So the lymphatic vessels themselves, they run right next to the arteries and veins. So here's an artery going in that direction, here's a vein going in this direction, and here's a lymphatic vessel going in this direction. And notice they've got these little valves. You can kind of see those two flaps there, and you can see it in this picture down here at the bottom. So there's not a lot of pressure. There's no heart pumping the blood in lymph. So the main way we circulate the lymph back is by muscle contraction. So if you take a step and we get a squeezing right here, sorry, that's kind of not easiest to see, you get a squeezing right there. The lymph, just like in veins, will get squeezed up here, and then it'll get caught by these valves. So right here is a valve, so the fluid won't be able to come back down. It will come back to only settle here, and then the next contraction gets squeezed further, and next contraction further and further. So every time you move, every time you take a breath, you're actually squeezing that lymph around. And that's why, I don't know if any of your parents complain about this. I always heard my parents complain about their feet swelling at the end of the day. So particularly people that have desk jobs and they sit a lot, you don't have a lot of muscle contractions and you don't squeeze this fluid out of your tissues very well. So your tissues tend to swell. They tend to collect this fluid. So moving around is a nice way to help with that circulation. So all of those vessels form this big map that you kind of see here, and this is just a generic map, guys. It's not super specific, but where we were in that big toe would be down here, and everything's going to kind of flow back up through here through muscle contractions, all the way up, all the way up, all the way up. And what you'll notice is right at like the groin here and here, you'll see these little bumps, and particularly in the armpits, things like that. So if you think about what we've got here, from all the extremities, all the fluid collecting this way, all the fluid collecting in from the arms, okay, and all the fluid collecting up from these legs are going to pass through all these lymph nodes, okay. So essentially, like they're strategically placed, pretty smart placement. They're checkpoints to make sure that there aren't any infections inside there. So let's look at a lymph node a little bit closer. So a typical lymph node kind of looks like this. Fluid would come in from a bunch of different directions, okay, into the lymph node. And inside here, it's going to be filled with all sorts of immune cells that are going to check. And we're going to talk about a lot of those as we go through here, B cells and T cells and so forth. And essentially what they're going to be looking is for weird antigens. Your cells have a certain combination of antigens, and a bacteria or virus is going to have weird antigens on the outside. And if we see some weird antigens, we hopefully will clean that out, destroy it before passing it on, because right this way is back to the bloodstream. So we don't want to pass an infection on that way. If we pass an infection in that direction, that means it's going to go into your blood. Now it's circulating to your entire body. So the lymph nodes are supposed to filter things out. Sometimes they filter out cancerous cells, and that's why you'll often hear about lymph nodes. You'll hear about women that, if we go back a couple slides here, women that have breast cancer. Let me get rid of clearing all this ink, okay? So with the breast tissue, the stuff drains through little lymph nodes, okay? So what they can do, if they have a tumor in one area, they can actually look at the lymph nodes that would drain from, that area would drain from, and they can actually remove those lymph nodes, look through those lymph nodes, and actually check for an infection, check and see if cancer cells move there. And that's generally a sign of metastasis, right? We've talked about the spreading of cancer. So that's one of the uses medically of lymph nodes, is to actually check and see whether something has had an opportunity to spread or not. The next organ that we mentioned is called the thymus, and the thymus is right here. I see this little kind of shape sitting on top here. So it kind of sits on top of the trachea, above the heart, kind of sits in that area. We'll see it really well when we open up the cats and look at the organs there in the chest. But the main job of the thymus is going to train your T cells. And we haven't talked about the T cells yet, but they are vital to your immune system, guys. So this is going to be a huge, essentially like a training camp for your most important soldiers in your immune system. So they come in here and they get trained, and really what they're trying to train them is to identify what's a normal antigen and what's a foreign antigen. So your body can actually tell your cells and recognize that that bacteria is not normal and it should be destroyed. And the last of those structures that we mentioned real quick is the spleen, and you'll see it kind of in purple down here, guys. So this is the spleen. We mentioned that it kind of sits, so here's the back of the body, here's the vertebrae, here's the front of the body, here's somebody's abdomen, right, in the front. We mentioned that it kind of sits over here around kind of near the stomach, okay? And what happens is blood flows in through all these red blood vessels, okay, flows in here into the spleen, and the spleen is like a huge lymph node where it's going to look at the blood for infection. So it's searching the blood for weird antigens. And the spleen, we mentioned in class, it swells up when you have blood-borne illnesses like mono, right? So mono will cause this spleen to swell up, just like a lymph node will swell up when there's an infection. And that swelling makes it vulnerable to kind of bursting, and that's why it's kind of dangerous for physical injuries during that period of time. So most of the time I refer to as the spleen as a lymph node for your blood. Okay, so fluid's going to flow into it, it's going to search it for normal antigens versus abnormal antigens and clean out infections just like your lymph nodes do for the lymph. All right, so this slide is actually a pretty complicated little slide, guys. It'll make a lot of sense by the time we finish the video. What I want to focus on right now is the fact that you have two big categories in here. We have this group that's called specific defenses, okay? And you probably want to define that if we didn't define it in class or you didn't define it in class. So a specific defense targets one antigen, okay? So it's going to target one weird marker on the outside of one virus or one bacteria, something like that. Okay. Our non-specific defenses, which I want to tackle first, is just your general defenses. It's things like, it can be things like your skin keeping things out, but we're going to kind of talk about this portion first here. And then we'll tackle the specifics. So we're going to start with this non-specific first. So here's the slide that we used in class that talked about non-specific defenses. So we have things like just physical barriers. Okay, sorry, helps if I don't hide the word. Okay, so we got things like physical barriers. So your skin, your hair, you have sweat that actually can work that way. When you pee, you wash out the urethra. You have secretions on your eyes that actually protect your eyes. So you have all sorts of physical barriers. Okay, then you actually have just the general phagocytes, so macrophages and neutrophils and all sorts of different cells, and their job is to swallow up anything that has weird antigens on it. So that includes sometimes cancerous cells. Remember, cancerous cells, because of anaplasia, will have abnormal antigens, so they will actually destroy and get rid of some cancerous cells before we ever see them. Okay. We then have these natural killer cells, and the natural killer cells are pretty cool. They aren't specific to one antigen, but they can recognize different antigens, and they don't swallow them up. What they do is they actually destroy the cell membrane. They release these little molecules that actually kind of create holes, and the cells just kind of fall apart. So they're a part of that. So they got these natural killer cells. Okay, interferons we talked about. The interferons, you keep it simple, guys. They interfere with viruses replicating. So if you have interferons in there, the viruses can't spread very well. Okay, and then the complement system. So the complement system is all sorts of different things. So they sometimes break down cell walls. They attract stuff. They stimulate inflammation. They work in a bunch of different ways from that standpoint. Then we've got like swelling, just a general swelling, right? So anytime we get swelling, if you say your fingertip gets infected and it becomes red and swollen, these are the reasons for that, guys. So we increase blood flow, it activates the phagocytes a little better, you get more capillary permeability, so more stuff is kind of coming into the area to help fight the infection. The complement system is activated. The local increased temperature makes the white blood cells a little more active as well. Right? And then the last one of the ones that we talked about was the fever. And fevers, remember, are intentional. They're caused by pyrogens being released. Okay, so you see that word pyrogens here. So remember, pyrogens being released by the white blood cells fighting the infection. So these cells here can release these pyrogens. Okay, and then those pyrogens go to that central thermoreceptor in your hypothalamus, and it just cranks up your thermostat. It's just like turning you from 98.6 up to, like, say, 101, 102. And the idea that because it mobilizes defense, it kind of stimulates the immune system. It can accelerate repairs. Remember, chemical reactions typically work faster when you heat them up. And then sometimes it can actually damage the pathogen. Some pathogens are sensitive enough to temperature that they'll actually be destroyed that way. Okay? So on the whole, the most important thing you guys got to know about this slide is that these are referred to as the non-specific defenses. And they're called non-specific because they do not target any specific antigen. They're just a general defense. So if I got infected, say, simultaneously with 3 different viruses and 2 different bacteria, you know, this one monocyte could fight all of those. Okay, natural killer cells could fight all of those. They aren't specific. Okay, what we're going to see now in a minute is the specific defense, and what will happen is there'll be a specific T cell and B cell to attack each of those different diseases that have been to the body. All right, so this slide gives you a big overview of the specific defenses. That's most important here. Okay, so we're talking about specific defenses here. And we have 2 categories. We got our cell-mediated, and we have our antibody-mediated. We're going to talk about this one first and this one second as we go through the process. Okay, so let's kind of jump into the big overview of how this all fits together. All right, so this is going to be the part 1 side. This is going to be the part 2 side over here. So we're going to focus on this part 1 side first here. So right away, when we get an infection, okay, right away when we get an infection, and it either the bacteria is found or a cell has marked itself. So remember that infected cells, when they get infected, they actually have a process where they mark themselves. They put that little protein on the outside, a chunk of the antigen, and they mark themselves as infected. Okay, so when that happens, it's going to be discovered by one of these killer T cells, these cytotoxic killer T cells, okay? And what they're going to do is they're going to multiply, okay? They multiply, and it takes a bunch of days for that to happen, guys. It's not a very fast process. And they circulate the body, and they find and destroy the infected cells, okay? And they do it a couple different ways. They have like a cell poison they can use. They can rip open the cell membrane. They can turn on a gene inside the cell that makes it essentially kill itself, okay? But that's one of our body's responses to these killer T cells. And so they're specialists. They're looking for the one specific antigen that has been found here. So this bacteria has one weird antigen. It is eventually found by those killer T cells, and those killer T cells then go and hunt that down. Okay? So there's a couple slides here that show you those steps. So this is showing you how the infected cell will mark itself. So this is just a normal infected cell. Okay? Virus comes in. As the virus taking over the body then makes this little structure and puts it on the outside. So it's identified itself as being marked. That's what we did in class when the kid held the little symbol over their head in class. Okay? So that's how a infected cell marks itself. Okay? This is going to show you how the cells become antigen-presenting cells and how they go and find the right T cells and B cells to actually go and fight the infection. Okay, so here's a white blood cell, right? Here's a phagocyte, okay, that has swallowed up this bacteria, and then it marks itself and then goes looking for help. So for the killer T cells, this is actually the slide that was important here. So, and I tried to mark them, like C, so you can kind of go back to this flow chart and be able to see, all right, so this C and C and C all corresponds to this slide. So this would be your visuals for that. And what we've got here is we've got your infected cell right here, kind of the tan, and here is the inactive cytotoxic or killer T cell, and then it becomes activated. And what happens is it goes and finds the cells and destroys them like this, okay? It also forms some memory cells, and those memory cells hang out for the rest of your life so that you can respond faster to the reaction, to the infection next time. So everything on the left-hand side here, all of this area is going to help us with cells that are already infected, okay? But the problem that still remains is any bacteria virus that's outside of cells is not going to be taken care of very well by this. We actually need this second part, which is part 2. And what's going to ultimately happen, guys, is we're going to make antibodies. And all of you guys have heard of antibodies. And that's the flow chart of how we actually make antibodies here. So the way it starts is you have a macrophage. So remember, macrophages are these big cells that swallow up a bacteria or virus. And then it becomes what's called an antigen-presenting cell. So that's the slide that I showed you here, right? So where it's swallowed it up and became an antigen-presenting cell. So once it's become an antigen-presenting cell, now it's got to go activate the helper T cells. Okay, so helper T cells are they're specialists. They only respond to one antigen. They're specific, and they've been waiting. Remember we talked about in class, they've been kind of waiting their whole life to be activated, and all of a sudden now they've been shown this one specific antigen, and then they will start to multiply. Okay, and once they multiply, then they will be able to go and activate the B cells. And the B cells really already have to be sensitized. They've already been kind of exposed to stuff. So you'll actually have this process where they have already seen the antigen a little bit and they're already ready to go, but this helper T cell is really what gives them the go signal to go fight the infection. Okay? Once they've been activated, they then change into what are called plasma cells. And plasma cells start dividing like crazy, and then they actually produce the antibodies. Okay? And so an antibody is this little kind of Y-shaped missile that's going to latch onto the antigen. We'll show you a picture of that in a second. Okay? So these antibodies are going to go fight the infection, and then what we end up with at the end is, again, some more memory cells. So we have these memory B cells at the end. Okay? So let's show you a couple of images that help you visualize that whole process. So here's the first part. Here's the macrophage. It is now an antigen-presenting cell. It's walking around with this antigen saying, hey, can anybody help me fight this infection? I don't really have a good image for like the antigen-presenting cell binding with the helper T cell, but this one slides right into the last part where we've got the sensitized B cells, because they're exposed. See all these little red dots? Those are supposed to be the antigens. They've seen some weird antigens. Okay. Now comes in. Here's our B cell, and here's the T cell that shows it. Here's the antigen-presenting cell, T cell, right? Shows it, and that's what really activates it. So those two cells binding together, that's what really activates it. and then this activated cell now starts to divide, become plasma cells, and start producing these antibodies, and then a few of them go off and they become memory cells. So what do antibodies actually do? Well, they latch on to the antigen. So these are antigens on the outside here, and you'll see how they're kind of like a lock and key fit here, so they just show you a nice simple circle. It's really pretty intense biochemistry that goes into it. But they latch on like that, and when they latch on, sometimes it destroys the bacteria virus, but more often it just kind of paralyzes it. This can no longer cause the body any damage, okay? So then all we got to do is wait for a big macrophage to come around, swallow it up, and eat the whole thing and get rid of it. So I'm going to try and use this slide to kind of put it all together, the whole physiology, but writing with these is a little bit tricky, so I'm not sure how well this will work out. Okay? So remember, we actually have two divisions of the immune system. We've got our non-specific, okay, and that includes things like your barriers and your skin and all those types of things. And remember that they aren't focused on one antigen, they're just focused on any weird antigen. So the same cell could attack a bunch of different infections, okay? What I'm most interested in, or the part that I find most fascinating, is the specific, okay? And the specific's got two different chunks. It's got our cell-mediated, okay? So cell-mediated immune system, right? And when we talk about the cell-mediated immune system, that's going to be either triggered by a bacteria being found, okay? So we got a bacteria or an infected cell. Okay, and remember, when a cell gets infected, what it does is so the little virus comes in, it marks itself. It puts the little weird antigen on the outside. I'm just going to put an A on the outside here for antigen, okay? So it marks itself. And when it does that, that's going to kind of activate our cytotoxic T cells, our killer T cells. So the killer T cell will come around here, and we'll just put usually they're T with like a little subscript K is how people usually draw them. What they'll do is they'll find this, and it'll get it activated and start multiplying, and it'll go search in the body for any more of that infection. Okay, and then some of those cells, so they're going to look for all the cells that are already infected. Okay, our other division is the antibody mediated. Okay, and the antibody mediated is going to start off with a macrophage. Okay, so we have our macrophage. Okay, that finds an infected cell, swallows it up, and takes off the antigen. So now you've got this macrophage here, and it's got this weird antigen on the outside. It's very similar to what we had over here. Okay, but it's actually a macrophage that's doing it. Okay, now that macrophage is going to go find our helper T cells. So that's usually marked with a T with a little H instead of the K over here. All right, now we have the H, so this is the helper T cell. And the helper T cell is going to go find our B cell. Okay, so B cells, remember, these have already been kind of sensitized, they've already been slightly activated, they're already, hey, something's wrong, but they don't really know what's going on or whether they should get involved. Well, the helper T cell binds to it and activates it, and this B cell will then start to multiply. It'll turn into a whole bunch of cells, and each one of those cells will then become what's called a plasma cell, and the plasma cell can make the antibodies, those little Y-shaped missiles that they're going to shoot out all over the place. Okay, so these are our different divisions, guys. We have our non-specific, which doesn't care what antigens, is weird antigen. We have our specific that is going to focus and attack one antigen. Right? In the specific, you've got the cell-mediated, which is going to tackle stuff that's inside the cells, things that are already infected, cells that are already infected, and it's usually these the killer T cells or the cytotoxic T cells that are going to recognize that and destroy that. And then we have the antibody-mediated, where it starts off with a macrophage, okay, which then interacts with a helper T cell to show it the antigen, gets that helper T cell all excited, the helper T cell multiplies, then finds the proper B cell, gets that thing all fired up, changes into plasma cells, which then start producing the antibodies. And this slide just kind of gives you an idea of the response time. So if you look here, like this is in weeks, it takes almost a week before you start really producing significant levels of antibodies, right? And that's kind of important to kind of remember here. So this whole process takes time. And that's why we talked about the fact that the memory cells are really important. If you have the memory cells, all of this stuff gets condensed, and we respond much, much faster. We would make antibodies much faster, we'd have plasma cells much sooner, we'd have all sorts of things happening at a much quicker pace. And that's why we talked about the primary response versus the secondary response. The first time you see a certain infection, all of this is pretty slow. The second time you see an infection, it is a really quick response. So if you have chickenpox for the second time, your body wipes it out probably before you ever get sick with it. And that's kind of the process and the way it's designed to work from that standpoint. All right, so one final overview to make sure that we understand what's going on. Everything's going to start with one weird antigen coming into the body, right? Non-specific defenses, we talked about lots of things, but we've got this little complement system that we kind of mentioned, and then the natural killer cells going and attacking the antigen directly. But what's really going to finish off an infection, that's one that's going to cure us of an infection, is going to be the specific defenses. And the specific defenses, remember, we have two different categories. On the left here we have the cell-mediated, on the right the antibody-mediated. So the cell-mediated, you get the killer T cells that are activated, and then they go and actually find cells that are destroyed. So they kind of multiply and divide, and they actually go and find the cells that are infected and destroy them. Okay. On this side, we've got the helper T cells activating on both sides. Okay, but in this case, the helper T cells are going to activate these B cells, which then turn into plasma cells, and the plasma cells start producing antibodies, and then the antibodies are what actually goes around this. These B cells don't have to go and find the infection; they just produce antibodies. They just flood your body with antibodies, and the antibodies will attach to the anything that's on the outside of a cell. Okay, and that's really important, guys, because on the left here we can tackle whatever's inside of the cells. Over here on the right we can tackle any pathogens on the outside of cells, and combined we can tackle any of the infection that you have in the body. All right? So hopefully this helps with a little extra reinforcement, a little review. Please come see me if you still have questions, guys. Have a good night.